Hi class, welcome to chapter 10 lecture part 2. Before we get started on the lecture, I have to apologize. I'm so sorry that this is only coming up onto Canvas on Friday afternoon. I'm teaching another class at the same time. I'm teaching a section of 205 this semester, and we're also in the genetics portion of that class as well. And so when I made the video for that class, for whatever reason, I got it transposed in my mind and I crossed it off the to-do list for this class. So when I went to reference like what I still had to do for the week, I'd accidentally crossed it off my list of things to do for 107 instead of 205. So I'm really sorry for the mix-up here. And I thank Mariana and Aileen for reminding me, sending me an email and asking me about the video. And if this ever happens in the future, please don't hesitate to reach out, text me, message me on that Pronto app or whatever, and I really appreciate it. And again, I'm very sorry. I will push back the due date for lab quiz 11, which is based on DNA to RNA to protein. I'll push that back until next week, and I'll give everyone that's already completed it another attempt. So if you want to go ahead and try that again, now that you actually have the lecture to kind of help you through it, I'm going to go ahead and do that. So if you have any questions, again, just let me know, and let's go ahead and get started with the lecture. So if you remember, we left off talking about how DNA is put together and how it is replicated and how those nucleotides are stitched together to make a strand of DNA. And so let's remind ourselves that DNA is made up of all these tiny little nucleotides, and we're going to we designate those nucleotides by the nitrogenous base that it's attached to. So remember, nucleotide has three parts. It has a sugar and a phosphate, and that sugar, that deoxyribose sugar and that phosphate never change. They're always going to be the same, and they make up the backbone of our DNA. And the nitrogenous base is that third part, and that's the part that varies. And in DNA, our nitrogenous bases are going to be adenine, guanine, cytosine, and thymine. And the way that those are put together literally makes up our DNA. Our DNA is two-stranded and it's twisted around itself. Now, there is another type of nucleic acid, and we're going to call that nucleic acid RNA. And I'm hoping that you can see, if you want to pause the video here and try to identify two major differences between DNA and RNA. Can you see what they are? Well, the first major difference, if you notice, the RNA also has four nitrogenous bases, and it has adenine, guanine, and cytosine, but instead of having thymine like DNA has, RNA has another nitrogenous base called uracil. So a very, very clear difference between DNA and RNA is that they contain different nitrogenous bases in their nucleotides. The other major difference between DNA and RNA is that DNA is double-stranded and RNA is single-stranded. So two big differences, double-stranded versus single-stranded, and the presence of thymine versus uracil. So let's talk about our end goal. Remember that the ultimate goal of DNA, remember that DNA, all DNA is, and really RNA also, are instructions. And they're instructions to make something. So what is that something? Well, it's proteins. And we've talked a lot about proteins already in this chapter. Proteins are really what do everything in an organism. All the enzymes that we use, all the enzymes that are in our cells and make things happen are ultimately proteins. And they have these really specific three-dimensional structures. And those three-dimensional structures dictate how that protein acts and what it's capable of doing. So the question is, how does that protein structure, what dictates that protein structure? Like, think about, if you see those beautiful, my niece is super into Lego, and she has these, like, of course she has to pick the most expensive ones. So I'm always stuck buying her these expensive Lego kits for her birthday, but she wants the ones that are like architecture Lego. Like, she has this one that's like an entire, like, she has one that's, I think, like the White House or something. So you can literally make like a little schematic of the White House out of Legos. And so when you're looking at, think of any kind of Lego structure in your mind, what ultimately causes that structure to look the way it does? Well, it's all the individual little Legos and how they're put together. And so if we want to think, if our quaternary structure are are many, many different kind of big Lego contraptions put together, right? Like if we stand back and we look at a scene of Legos, and it's like a little Lego town, and we have cars and buildings and city streets and so on, that would be considered our quaternary structure. When we put proteins together to form something that's functional, we call that the quaternary structure. Below that, one step below that, is going to be the tertiary structure. And these tertiary structures are kind of like the individual strands of proteins and how it folds on itself based on, of course, the secondary structure. And the secondary structure are maintained by hydrogen bonds. So the way that we snap those Legos, kind of the way that the Legos, I should say, kind of fit together after we put them together. So if you snap two Legos together and then you snap more Legos together, and the way that those two groups fit together would be kind of considered our secondary structure. And there's two kind of patterns here. We can have an alpha helix, and this is kind of similar to DNA where we have like a twisted protein structure. This is actually what we see in our hair, by the way. Our hair is made up of keratin, which is a type of protein. And the way that that protein kind of twists on itself gives our hair its texture. The other major type of secondary structure is a beta pleated sheet. So we can kind of think again, so all of these, like these three kind of larger, how would I say, these three larger patterns that we see in proteins, secondary structures, tertiary structures, and quaternary structures, are all ways that proteins kind of self-fold. So again, if we're thinking of Legos, how the major structures of those Legos are look and how they're put together. But ultimately, it goes back to a very, very simple thing, and that is simply the sequence of all of those monomers that make up a protein. If you remember, the monomer of a protein is an amino acid. Another term for it, we sometimes call it, I believe your book sometimes uses this, is a peptide. So to me, peptide and amino acids, they really mean the same thing. So sometimes people call them peptides, sometimes people call them amino acids. I usually refer to them as amino acids. And it's this primary sequence of amino acids that ultimately determines, so this determines a secondary structure determines tertiary, and tertiary determines the quaternary structure. So it's truly that sequence of one amino acid after the next that will determine how proteins look. So DNA gives us those directions. DNA actually gives the directions to RNA, and RNA uses those directions to make a string of amino acids. And if you look at this string of amino acids, the first, sure, why not? Let's say this is the first one. It's this PHE stands for the amino acid phenylalanine. The next one is valine. The next one is histidine, and then valine. Whoa, what happened? Sorry about that. Valine, and then alanine, and then valine again, asparagine. Why is my screen is acting weird. And so on. So it's these string, there's about 20 amino acids that make up all of our proteins. And so somehow DNA has to direct this assembly. How does phenylalanine know to be first? How does valine know to be second, and histidine third, and so on? Well, that's the whole idea behind molecular biology. So let's get into this. So again, this idea behind molecular biology and how DNA directs the synthesis of RNA is through an intermediate that we know as RNA. So RNA and DNA are both types of nucleic acids, and DNA is going to get transcribed into RNA using a process called transcription. So transcription is the process of making DNA into RNA. Notice that it's happening in the nucleus. Here's our nucleus. And that RNA transcript, this little pink squiggly here, is going to go out into the cytoplasm, and it's going to get translated into a protein. So we have two major processes here that we need to talk about, and that's transcription and translation. Transcription is the process of turning DNA into RNA, happens in the nucleus, and translation is the process of turning RNA into protein. This happens in the cytoplasm. So let's remind ourselves that ultimately what we're talking about here is gene expression. Remember that Mendel and some of the other, the other random scientists that we've talked about in some of these experiments, they all described a gene as being this discrete heritable piece of information. And that's exactly what a gene is. So we have our chromosome here, we have a duplicated chromosome, and if we were to unwind that chromosome and look for a gene, we actually see those genes in the DNA itself. So genes are located in DNA. They're very specific sequences of DNA that code for a protein. So sometimes you hear proteins being described as a gene product. That's a very common way for researchers to describe proteins, because ultimately the product of a gene, the way that a gene is expressed, is through a protein. So let's look at exactly how this is happening. Remember that DNA has a specific orientation. We can have a 5' end and a 3' end of DNA, and then we have those strings of a sequence, I should say, of nucleotides, A's, C's, T's, and G's. Those sequences are going to be turned into a complementary copy of RNA. So this pink copy, notice, is RNA. And how do you know this? Well, one, you know it's RNA because we say that. We say it's RNA. But if you look at that string of pink nucleotides, how do you know that's RNA? Hopefully you recognize it's RNA because it has U in it. It has that nucleotide uracil in it. And that should be a good indicator that this is a string of RNA. And we're going to call this string of RNA a special name. We're going to designate it as a message, right? So the M here, mRNA, stands for messenger RNA because it's sending a message from the nucleus where the DNA is. Why is my screen I hope you guys don't see that. Where the DNA is, out into the cytoplasm to eventually translate this polypeptide, this protein. So notice that the complement of A is U, the complement of C is G, G is C, and so on. Right? So we know that this is a complementary copy. It still follows those rules of complementary nucleotide base pairing, except that in RNA, A is not going to pair up with T because T doesn't exist. A is going to pair up with U. So to continue kind of our information flow, this messenger RNA is going to go out into the nucleus and eventually code for a particular kind of amino acid. And it does this through these I dropped my pen. It does that through these very specific sequences called codons. And a codon, I think I have this in another slide, but a codon is a triplet of 3 nucleotides at a time. So it's kind of a reading frame for our cell, all life cells, all life forms. Again, every single life form on Earth does this. We have this in common with bacteria and fungi and what are my other go-tos? Flamingos and ducks and so on. So every living thing on this planet has this very specific code of triplets, triple sequences, three nucleotide sequences that we're going to call a codon. And that codon codes for a specific type of amino acid. So UUU would code for this amino acid, GGC is going to code for this amino acid, and so on. So let's look at these codons in a little bit more detail. So again, a codon is read in frame. So that means that they're read 3 at a time, and they're always a repeat, or I shouldn't say a repeat, but a set of 3 codons. And so that triplet has a special name and it's called a codon. And notice that codons are in RNA. We could potentially say that there's some type of complementary triplet in DNA, but really when we're talking about codons, we're talking about RNA, because RNA, that messenger RNA, that string of RNA that's going to go out into the cytoplasm to code for a protein, is really the code, right? DNA is really the instructions. It copies those instructions onto a code called messenger RNA, and so our codons are on that RNA. So As you can see here, this is one example. I believe this is the example that's in your book of a table that actually tells us what codons code for what amino acid. So, I mean, think about this. If you're translating a language, right? So let's say that we need to translate English to German, for example. We can go to a dictionary and we can look up a word in English, and that dictionary will tell us what that word is in German, for example. And that's exactly what this chart does for us. It gives us every possible combination of those 3 nucleotides. Remember, or I'm sorry, those 4 nucleotides. Remember, we have 4 possibilities: U, C, A, and G. And these 4 combinations have to be in a triplet. They can only be at 3 at a time. So we see every possible combination on this chart. And this chart tells us what combination of letters are going to lead to what amino acid. So, for example, UUU and UUC code for the amino acid phenylalanine. Any of these CU combinations, CUU, CUC, CUA, and CUG, are going to code for the amino acid leucine, and so on. So this is a really handy chart that you can always reference when you have to make an amino acid from a messenger RNA sequence. There's a couple things that I want to point out in this chart. First of all, the amino acid, the codon AUG is our start codon, and we'll talk a little bit more about what a start codon is later. But a start codon is the signal for the ribosome to assemble and start making protein. And so this is the only one, AUG. I remember it as a start codon because AUG sounds like August, and school always starts in August. So that's how I remember the start codon. The ribosome won't start making protein until it sees that start codon AUG. Now, on the other hand, how does the ribosome know when to stop? When does it stop making protein? Well, it's going to stop when it runs into any of these 3 specific stop codons: UAA, UAG, and UGA. So we have one very special start codon. We have 3 unique stop codons, and all the rest are going to code for those roughly 20 amino acids that make up our proteins. Notice that our amino acid code, our codons are redundant. In other words, multiple codons will code for the same amino acid. So you can have any of these 4 codons, and they'll still code for proline, for example. So this is kind of what I was talking about in the previous slide. So nearly universal. In other words, again, bacteria, fungi, flamingos, any living thing on this planet uses this same code. I think there are a couple of really interesting examples of archaea. Remember, those are the very odd prokaryotic life forms that occupy very odd niches that have some small variation in this code. But other than a few very interesting instances where there's some very small changes, everything on our planet uses this code. I think it's a really cool, remember, our theme for biology is unity, unity and diversity. As diverse as our life form, as diverse as all of the life forms on this planet are, we all use the same code. And I think that's a really beautiful thing. It's really cool to think about that. As diverse as all the different species are, we still manifest life in the same way. That's really cool. And again, it's redundant because we can have more than one amino acid, or I'm sorry, I keep transposing this, more than one codon code for, I just want to make sure. It kind of looks like I'm not recording anymore. I'm just going to escape out of this really quick and make sure that I am not losing you guys. And I'm still recording. Good. Okay. Let's go back to the slideshow. Come on. Okay. All right. So it's redundant. So this term redundant means that we can have several different codons code for the same amino acid. But notice that no codon will code for two different amino acids. So it's kind of a one-way street. We can have multiple codons coding for one amino acid, but we can't have a codon coding for two or more amino acids. In other words, CAU will always encode for histidine, nothing else. All right. So if we have our strand to be transcribed, our DNA, remember DNA is always double-stranded, our messenger RNA is going to be made in the same direction as new DNA is made. In other words, it's always going to go from 5 prime, the 5 prime end of our nucleic acid to the 3 prime end. So if we look at this above sequence, we are transcribing this strand of DNA. And this strand of DNA is going to be called the template strand. So notice that it's complementary. T is going to code for A, U, CG, TA, TA, and so on. So we're building our RNA strand. Notice that we have our start codon here. And then if we were to reference AAG, let's go ahead and look back at our little table here. If we find AAG, so here's our A, here's our A, AAG codes for the amino acid lysine. So our next amino acid is lysine. And then our next amino acid, remember, here's our triplets. I'm sorry, I hope you're not seeing my screen blanking out like that. Our next triplet is UUU. So what does UUU stand for? UUU stands for phenylalanine. There we go, our phenylalanine. And then our last codon is our stop codon. So this particular amino acid has this particular protein has 3 amino acids, which is impossible because most proteins are, oh my goodness, they're thousands and thousands and thousands of amino acids long. But we're not going to make you do all those translations. That would just take too long. And you get the idea. All right. So I think I mentioned this in the previous slide. Messenger RNA, right? Our message is always made in a 5 prime to 3 prime direction. So here's if our this pink strand is our newly made DNA. Here's our 5 prime end, and we're always adding new nucleotides to that 3 prime end. So we're always reading the DNA in a 3 prime to 5 prime direction. Orientation is a tricky thing to kind of wrap your mind around with transcription, with replication too, and transcription. Okay, let me go ahead and get all this out so we can talk about, whoops, let me go one, there we go. All right, so when we think about the process of transcription, so this schematic is telling us, is kind of a visualization of how transcription occurs. Here we have our DNA, and every time we start a process, if you want to think about the process of replication, for example, it's going to have a beginning and a middle and an end. And if you think about let's just do a quick review here. If you think about replication, our beginning was when the enzymes would assemble at our origin of replication. Remember, there's lots and lots of origins of replications that are going to begin all throughout our chromosomes. And the middle part would be elongation, as the leading and lagging strand are made and we're adding nucleotides to those complementary strands of DNA. And termination would occur when those leading and lagging strands meet the other origin of replication, right? They are continuing to elongate until they meet up, and DNA ligase will come in and stitch those pieces together, and we're done. So we always have a beginning, a middle, and an end to every process, and transcription is the same. Transcription is going to have an initiation, an elongation phase, and a termination phase. And so how do we know? How do we know where to start? How do we know where to end? So let's talk about where we start in DNA. DNA will have a very specific sequence hidden inside of its hidden inside of its sequence of nucleotides called a promoter sequence. And a promoter sequence is a very specific sequence of DNA that tells an enzyme called RNA polymerase to come and bind to it. So RNA polymerase is going to land and be able to bind to DNA in this specific area called a promoter sequence. And RNA is going to start to move down the DNA in a 3' to 5' direction to elongate that pink, right here's our little pink messenger RNA strand. So we have elongation is occurring, and elongation is fairly straightforward. We're simply adding RNA nucleotides to the 3' end of the messenger RNA. But how does it know where to stop, right? Why doesn't RNA just, the RNA polymerase enzyme, why doesn't it just keep going? Well, it's going to reach another very specific sequence of DNA called the termination sequence. And the terminator sequence tells the RNA to stop and detach and fall off. So the RNA sequence is going, the RNA polymerase is going to fall off. We now have our strand of messenger RNA and transcription is done. We've made a copy of DNA. So let's practice this. So here we have a string of DNA. Notice that the 3' end is here, the 5' end is here. And we want to make a messenger RNA transcript from that. So we're actually going to do transcription, and let's go ahead and do translation since we have the codon chart here as well. So go ahead and pause the video and see if you are able to do this, this sequence. So I'm going to go ahead and do it here. Hopefully you already have the answer and you can just kind of go along with me. Remember that messenger RNA is always going to be made in a 5' to 3' manner. So if we have C's, C is going to be, the complement to C is G, T is A is U, G, C, U, A, U, U, C, G, G, A, G, A, U. All right. So now we need to use this messenger, sorry, that's kind of sloppy. Now we're going to use this messenger RNA as a template for making amino acids. And remember, we always start with our start codon. So we're searching for AUG. So we're kind of going along and we see it. Here it is. And then what I would recommend you do, this is what I always do when I'm trying to do this, is I immediately make all of my triplet codes. So here's my second one, here's my third one, and there's my fourth one. So my first triplet, AUG, stands for methionine. The abbreviation is MET. The second one, CUA, CUA stands for leucine, LEU. AUG, CUA, UUC. UUC is phenylalanine. All right, so let's do, what did I say? CUA. CUA is here. UUC is here. And our last one, GAU, is going to be GAU is going to be asparagine. Or no, that's not asparagine. Is that asparagine? It might be aspartic acid. Let me look. Aspartic. I forget what my, I'm rusty. I forget what my abbreviation. So this could stand for, I'm pretty sure this stands for asparagine. But it could stand for aspartic acid. I forget. Okay, so there's our 4 amino acid sequence that we got from our original DNA message. Methionine, leucine, phenylalanine, and aspartic acid. Let's try it again. Here's our next sequence of DNA. Again, pause the video, try to do this yourself. It's on your quiz. It's going to be on the test, I promise. Get some practice in here. just make sure I did this right. I don't know why it doesn't feel right to me. G is C, T is A is U, C, G, T, A, G, C, A, U, G, C, A, U, T, A, G. Okay, good. A, U, G, C. Okay, I guess that's right. So A, U, G, remember, is always going to be our start codon, methionine. G, A, C. G, A, C is that pesky asparagine or aspartic acid. I'm not sure which. GAC, UUC, phenylalanine. And finally, UAA, oh, it's probably going to be our stop codon, is our stop codon. So in this one, we only have 3 amino acids, and this is in comparison to this one where we had 4 because we didn't have a stop codon yet. We just kind of ran into the end of our sequence. Cool. Okay. So before we can even, I translated the protein there just because we had the codon chart there, and I just wanted to kind of show you how that works. But let's back up a minute because we're not actually there yet. We got to get to the ribosome first before we talk about translation. And in order to get to that ribosome, we need to get out of the nucleus. Messenger RNA, remember, in eukaryotes, because eukaryotes have a nucleus, needs to get out of the nucleus. And in order for that to happen, several things have to occur. So before we talk about getting out of the nucleus, let's talk about processing the message, the messenger RNA that is a result of kind of copying that DNA. Now, messenger RNA contains what we're going to call, well, I shouldn't say, yeah, okay. So DNA and RNA contain these pieces of DNA called exons and introns. Now, the way that I remember this is that exons exit, they leave the nucleus, and introns stay in. So what we're really looking for ultimately are these exons. So the exons are what's going to make up our messenger RNA sequence that gets to leave. Introns are these large stretches of DNA that get removed from the messenger RNA. Now, I remember learning about introns when I was in, maybe even in high school, but in high school, college, and I struggled with these because I don't get the point of it. Everything up to this point, like, oh, there's a code, we have to translate it, and this, codons and so on. Everything made sense up until these introns. And really, introns, to a lot of people, to this day, we're still studying what introns are because there really doesn't seem to be much of a point to them. So there are some interesting examples of alternative splicing, and there are some intriguing hints about what introns do, but nobody really understands what the point of them are because we go through all the trouble of transcribing them, right? To make, when we make DNA into RNA, we go through a lot, because this takes energy. Think about all the ATP, what we just got done talking about. Remember, in metabolism, it was all about making ATP. Why do we need ATP? Well, to do stuff exactly like this, to make DNA into RNA and to make RNA into protein. That's an incredibly time and energy consuming event. And so it always troubled me, like, why would we spend all of this energy to make introns just to cut them out? And it's kind of one of the mysteries of molecular biology. Again, there are some kind of intriguing hints that some scientists are studying now about what the point of them are, but really nobody really knows what the point of them are. But they get removed, and it's part of processing this messenger RNA. And the exons are stitched together to make this mature messenger RNA transcript. And a couple other things happen to this messenger RNA transcript. A cap is going to be added to one end, and a tail. We're just going to call them a cap and a tail. They're going to be added to each end. And now this mature messenger RNA transcript that has all of the introns removed, we have a 3' cap on one end and a 5' tail on the other end. It is now ready. It's stabilized, and it's protecting the RNA from degradation. And now that RNA can get out of the nucleus and go into the cytoplasm. So once the the message leaves the nucleus, it's going to go into the cytoplasm, and it has to find a ribosome. A ribosome is a little very complex, very fascinating piece of machinery that's made up of, actually it's made up of some RNA and some proteins, and its job is to turn this message of RNA into a protein. So remember, our first step is that transcription, and then our second step here is translation, right? So we have transcription occurring in the nucleus, and now we have translation occurring in the cytoplasm. So I'm going to go over this schematic, the lower part of this slide here in detail. So before we talk about the process of translation, let's talk about some of the major players. We already know that one of the major players is the code itself, that message or the messenger RNA. The other important player is transfer RNA or tRNA. And tRNA, if the messenger RNA is the code, tRNA is the interpreter. It's going to take the code of those codons, those 3 nucleotide sequences, and it's going to translate it into another language of amino acids. And so tRNAs have this very, very complex 3-dimensional structure that looks like a mangled DNA molecule kind of, but it's really made up of RNA. And so we usually use this kind of simplified version of what a transfer RNA looks like that points out its anticodon here at the bottom and the site where the amino acid attaches at the top. Those are the 2 important pieces of the transfer RNA. So the part that's going to recognize our codon and the part that's going to recognize our amino acid. So let's look at how this works. So before we talk about how this works, let's talk about our third major player. So our first one, remember, is our messenger RNA. Our second, I'm sorry, I don't know what's going on with my screen. My second major player is my interpreter, my tRNA. And then my last important piece of the puzzle is the ribosome. So the ribosome is the site where everything is going to come together. Notice that we can see our messenger RNA. Here we see our tRNA. And then this big brown thing in the back is our ribosome itself. And so you can see that that messenger RNA is going to get fed right through the middle of this ribosome, and the transfer RNAs are going to recognize those little triplet sequences here to translate that message into amino acids. So let's look at how that works. So again, let's think about our process. Remember, every process has a start, an initiation, an elongation phase, and an end phase. And so we probably already know where the start is in translation. Well, it probably starts with our start codon. Notice our little AUG, start codon. So when our messenger RNA is going to attach itself to one piece of the ribosome. So this brown piece here at the bottom is a smaller ribosomal subunit. And that attachment is going to attract our first transfer RNA. Notice that it's recognizing that start codon and it's attached to the amino acid that AUG codes for, which, if you recall, is methionine. This sequence recruits the large ribosomal subunit. So I've always thought of, it's always something that I've had in my head, but I always think of the ribosome as kind of like a hamburger bun. Like, so this is the bottom hamburger bun, the small subunit, and the top of the bun is the large one. And only when those 2 come together do we have we truly started protein translation. So now we have full assembly of this protein making machinery. So we have all 3 pieces in place. We have our messenger RNA going, which is our code. We have our interpreter, our tRNA, and then we have our ribosome fully put together, the large unit and the small unit. Notice that this ribosome has 2 kind of major sites. It has a P site and an A site. So let's talk about what the point of those are. So we've initiated protein we've initiated protein translation. So let's talk about elongation. So how do we start adding amino acids to this polypeptide? Well, we're going to have new amino acids come in. They're always going to come into that A site. And that's going to allow this amino acid to attach to that growing chain. And as soon as that happens, the tRNA that's in the P site is going to leave. The A site is going to move over one. And now we're going to have a new one. So the A site moved over. And now we're going to have a new a new tRNA come in, which we see here. So it's kind of a cycle. And you can see that every time we go through this cycle, we're going to add more amino acids to our growing chain. And remember that proteins are thousands and thousands, sometimes tens of thousands of amino acids long. So of course we already know that it's going to terminate when it finally reaches a stop codon. So that stop codon doesn't actually code for an amino acid, and that basically kind of confuses the ribosome. It doesn't, no transfer RNA is going to come in, and that causes the ribosome to pause and it falls off. And we now have our polypeptide. So again, remember, we have three major stages for every process. In translation, we have initiation, we have elongation, and then we finally have termination. So initiating is starting at the start codon. We elongate as we move down that messenger RNA, and then it stops when we reach a stop codon. All right, so now that we know how DNA is made into protein, let's talk about how DNA can be mutated. In other words, can that code be corrupted in any way, right? We know that in computers, for example, the binary code that makes up our computers can get corrupted by taking on a virus, for example, or some injury to our computer, or as our computers get old and don't work as well, we can have mutations in that binary code accumulate and our computers stop working as well. Well, that also happens in our DNA. As rare as it is, we can have a mix-up in our DNA where one nucleotide is substituted for another. And these are errors, and these will result in, so this is an error in our DNA sequence, and that can result in errors in normal cellular processes. So, and then I've always, because I consider myself, I guess, more of a molecular biologist. I always think about, when I think about mutations, I always think about mutations in the sequence of these nucleotides. However, there could also be chromosomal mutations. And so aberrations in our chromosomes, for example, a piece of our chromosome could be missing, or we could have an odd number of chromosomes. So we could have an extra chromosome be present in our nucleus. So there's chromosomal mutations, but the types of mutations that we're going to talk about are mutations in the actual gene itself. So point mutations are gene mutations that usually involve taking out one nucleotide and substituting it for another. So if we look at a normal piece of DNA, let's say this DNA codes, the sequence is CTT. Remember, that will code for a messenger RNA of GAA, and that will in turn code for an amino acid in a protein called glutamine, or glutamate, I'm sorry. Glutamate? No. I think it's glutamine. Yeah, it's glutamine, normal hemoglobin. So in glutamine. Now let's look at a mutation. So here we have a single mutation. This T was accidentally turned into an A. So now our codon no longer reads GAA, it reads GUA, and that now codes for the amino acid valine. Now, you wouldn't think that a single change, a single mutation in a single nucleotide sequence, because remember, we have, what do we have, like 3 billion base pairs? How many base pairs do we have? I should Google this. Where's my phone? I want to say we have like I feel like we have like 3 billion base pairs in even one of our, just in a single cell, we have like 3 billion base pairs of DNA. So you wouldn't think that a single nucleotide change would make that big a difference, but this makes a huge difference because it turns out that changing this single amino acid will result in sickle cell anemia. So this is one of the most well-characterized genetic disorders that are currently being studied, and it's all due to this single nucleotide substitution where we have an A instead of a T. So let's look at these different types of mutations. So we can have three different types. We can have a substitution, which is what we just talked about. We could have a deletion, or we could have an insertion. So a substitution is exactly what we just mentioned. So let's see what's different here. Instead of a G, we have an A. And that's going to result, instead of a glycine, that's going to result in the amino acid serine. So we could have a substitution. So that's where one nucleotide substitutes for another. Now, on the other hand, we could have a deletion occur. In other words, this particular nucleotide is completely taken out. What that does is it shifts all the nucleotides down one, because remember that our codons are in groups of 3. So instead of UUU, which is what it's supposed to be, it's now UUG because everything shifted down 1. This is really bad. This always leads to something bad. This completely screws up. Because I mean, think about reading, like when you're reading like your textbook, which I know you guys do every day, right? You're reading your textbook. Imagine if you removed a single letter from a word. Removing that single letter wouldn't be so bad, but if you moved all the other letters in your textbook over one, nothing would make sense. It would be all nonsense. And that's what happens in a deletion mutation. It also happens in an insertion. So an insertion is really the opposite of a deletion. We're going to add an additional nucleotide in here. So instead of it being UUU, now we're going to insert that G, and that's going to completely shift everything over as well. And so all that to say that substitution mutations can be bad. For example, sickle cell is a great example, but that's a really extreme example. Typically, substitution mutations don't lead to anything terribly bad. But nucleotide deletions and nucleotide insertions, these guys are bad news. Because they change the reading frame. Every single amino acid after that insertion or deletion is going to be screwed up. So what are the outcomes? So we have our 3 types of mutations: substitution, deletion, insertion. Now let's look at the outcomes. So we can have 3 different outcomes. We can have silent, nonsense, or a missense type of mutation. So if our no mutation looks like this, and it eventually leads to an amino acid called lysine, it's possible that we could have a mutation. Notice that here we have a base. It looks like we're basically doing a base substitution. Here we're going to substitute that T for a C. That still codes for the amino acid lysine. So this is considered a silent mutation because we aren't going to see any change in our polypeptide or our amino acid sequence. On the other hand, let's talk about missense mutations, and let's not even worry about these 2 terms, conservative and non-conservative. In fact, let's just not even look at this one at all. We can just leave it out. So in a missense mutation, we're actually going to change the amino acid. For example, instead of coding for lysine, we're now coding for arginine. So we're going to call that, a mutation that leads to a different type of amino acid, we're going to call that a missense mutation. Our last mutation is probably the worst mutation, and it's called a nonsense mutation. It's possible that this mutation can lead to mutate. So instead of AAG, we see UAG. And UAG is unfortunately a stop codon. So what that's going to do is cause the ribosome to stop translating the protein. And instead of translating the full protein in its entirety, we're going to stop maybe halfway or 25% of the way through translating that protein. And now we have a shortened protein. So imagine how good half a Lego would do you, right? Like, and not the good half, like not a half that's still functional, right? Like think about cutting it like diagonally or something, right? It wouldn't do any good and it wouldn't be a functional Lego anymore because it doesn't work. It doesn't fit in with all the other Legos. And ultimately, that's really what we're doing. We're making these really, really complex Legos that have to fit together just right in order for us to maintain homeostasis. And if we have a nonsense or a missense mutation, that's not going to happen. Okay. Insertion, deletion, frame shift. So yeah, so here's just a nice example of how we can lead to frame shift mutations where we see the entire frame of the codon shift. So here we have an insertion mutation. So we accidentally, when we were, say, replicating our DNA, we accidentally inserted a T in there, and that shifted all of our nucleotides down one. And now we have, instead of having our correct amino acid sequence, everything that comes after that now is going to be incorrect. However, if we insert, if we substitute, so we don't, let me just make sure I'm understanding what's happening here. Insertion, mutation. Oh, you know what? We don't have to even worry about this because I remember just a quick heads up, that's not on the test. So let's not, that's a different type of mutation that we didn't talk about. So I'm going to leave that out. Don't even worry about it. You can cross it off of your PowerPoint and let's not talk about this. So here we had a nucleotide insertion and it changed the amino acid sequence downstream of that insertion. So that's all we need to know from this particular frame or this particular slide. I think we're getting close to the end. I think this is our last slide. What type of mutation here is shown? So here we have normal gene, CCG. Our mutant gene is CTG. So hopefully you can see that we're simply substituting a T for a C. So this would be a substitution mutation that is going to lead to a different type of amino acid. So that's going to be a missense mutation. So this is a substitution mutation that's leading to a missense mutation outcome. Oh, cool. One more. Let's see. Normal gene. Oh, I like this one. Yeah. So go ahead and try to transcribe and then translate this gene. Whoops. Sorry. Okay. So pause the video and I'll do it while you're doing it. Just don't peek, don't cheat and look at me. T is A, U, G, 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 C, G, G. Glycine and UGG. What is UGG? GG is tryptophan. Okay, so the normal gene, when transcribed into our messenger RNA, looks like so: AUG, GGC, UGG. And if we track those codons to their amino acids, we can see that we get a 3 amino acid protein, methionine, glycine, tryptophan. However, in this one, we still get methionine and glycine, But here we have a substitution, right? We're substituting this T for a C here, but that leads to a stop codon. So here we still have a substitution mutation, but this is going to lead to a nonsense mutation because it leads to an early stop codon. Okay, I think that does it for excuse me, I think that does it for this lecture. We learned about the structure of amino acid, we learned about the differences between DNA and RNA. We learned about the structure of proteins and how that initial amino acid, that primary sequence of amino acid, determines the structure of proteins. And we learned about two major processes in this flow of information. And of course, that's transcription and translation. And then finally, we learned about mutations, the different kinds of mutations that can occur. We have substitutions, deletions, and insertions, and the different outcomes that can occur from those mutations. And we had a silent mutation can occur, a missense mutation, and a nonsense mutation like we see here on this last slide. And that does it. And I think this is our last lecture before the Unit 3 exam. So good luck on that exam. Again, as always, email me or contact me if you have any questions. Discard those guys and let's get rid of... Ooh, an hour. Good Lord. Congratulations for sticking through that one. Whew.